Okay, so welcome back to this next video in which we are discussing the measurement of intracellular calcium concentration using the Fluo-4 dye. Okay, so at the moment we're in the process of discussing the motivation for wanting to know the intracellular calcium concentration at all, and generally it's because you want to study the effect of receptors on the intracellular calcium concentration. So you want to see how activating a receptor on the surface of the cell membrane with its ligand can then cause an increase in the intracellular calcium concentration. Okay, so you want to study calcium signaling basically. Right, so the example of a receptor that we are looking at is the M3 muscarinic acetylcholine receptor, the endogenous ligand for which is acetylcholine. Okay, so we've discussed that when acetylcholine binds to that M3 muscarinic acetylcholine receptor, you'll get a change in conformation in the M3 muscarinic acetylcholine receptor, specifically in these three intracellular loops that we have on the cytoplasmic side of the membrane. These will change conformation and make available a binding site for the alpha subunit of the heterotrimeric G protein. Now, it won't just bind to any old alpha subunit of the heterotrimeric G protein. Instead, it has its preferences, and the M3 muscarinic acetylcholine receptor likes to bind to G alpha Q/11 family alpha subunits. Okay, so if you've got a heterotrimeric G protein involving one of these alpha Q/11 family alpha subunits, then the M3 receptor, once activated, will be able to bind to that. It doesn't really matter what the beta and the gamma subunits here are. Okay, right. Then, once the alpha Q/11 subunit has bound there, and I should say that the beta-gamma subunit, although this isn't involved in actually binding to the intracellular loops of the G protein coupled receptor itself, it being bound to the alpha subunit is essential for this alpha subunit to be able to bind to the G protein coupled receptor, and that becomes important in a moment. So once the alpha Q/11 subunit is bound to the intracellular aspect of our G protein coupled receptor, which for an example could be the M3 receptor, what happens is it loses its affinity for GDP. The GDP can then unbind from the alpha subunit, and then it will be replaced by a GTP molecule from the cytoplasm. When GTP binds to the alpha Q/11 protein, the alpha Q/11 protein changes conformation and adopts the on-state conformation, and in this conformation it will cleave away from the beta-gamma complex, so the beta-gamma complex will be released. Okay, so it will go off still attached to the underside of the phospholipid bilayer, but no longer attached to the alpha subunit, and it will bind to other targets and cause downstream signaling cascades. Okay, but for our purposes, the more important subunit is the alpha Q/11 subunit, which now has GTP bound, and because it has lost the beta-gamma complex, it can no longer bind to the intracellular loops of the G protein coupled receptor, so it too is released and goes off, still attached to the underside of the phospholipid bilayer. So, what does the alpha Q/11 family alpha subunits of heterotrimeric G proteins interact with? Okay, well, the main target is Phospholipase C enzymes of the beta family. So there are many different families of phospholipase C enzymes. Okay, one of the families of phospholipase C enzymes is the phospholipase C beta family. Okay, so let me just write this down here. So, there is a large family of enzymes known as the phospholipase C enzymes. In total, there are 13 different phospholipase C enzymes. Okay, and they are grouped into 6 families. Okay, so there is the phospholipase C beta family, and for short, phospholipase C is often abbreviated to PLC. Okay, and I will show you what phospholipase C enzymes all do in a moment. Okay, so PLC is short for phospholipase C. So then, firstly, there's the phospholipase C beta family, and this contains 4 different genes which code for slightly different proteins, but which are all similar enough that they're all going to be activated by the same thing, and they're all going to catalyze the same thing. Well, all phospholipase C enzymes, all 13 of the things, and I should say this is 13 different genes rather than 13 different enzymes. There are different splice variants of these genes. Okay, so phospholipase C beta family enzymes, there are 4 different genes under here. Okay, so there is phospholipase C beta 1, phospholipase C beta 2, and phospholipase C beta 3 and 4 as well. Okay, and before I realised that I needed to replace enzymes with genes, what I was about to say is that all the phospholipase C enzymes catalyze the same reaction. Okay, but what they are regulated by differs. So the different families are all regulated by the same thing, basically. Okay, to give some other families, there is also the phospholipase C gamma family, which contains 2 genes, the phospholipase C gamma 1 and the phospholipase C gamma 2. There is also the phospholipase C delta family, which contains 3 members, phospholipase C delta 1, phospholipase C delta 3, and phospholipase C delta 4. Okay, continuing on, there is also the phospholipase C epsilon family. There's only 1 member in the phospholipase C epsilon family, known as phospholipase C epsilon 1. Then we're going into fancier Greek letters, so then there is the phospholipase C eta family, which contains 2 members, the phospholipase C eta 1 gene and the phospholipase C eta 2 gene. And then finally, there is also the phospholipase C zeta family, which just contains 1 gene, the phospholipase C zeta 1 gene. Okay, so these are the 13 different genes. Let's just count them up to make sure I've got them all. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, so those are the 13 different genes for phospholipase C enzymes. Now, many of them do have multiple splice variants, which means that there's more than 13 different types of enzymes. Okay, but those are the genes for the enzymes. Right, so the ones that we're going to be interested in is this phospholipase C beta family. We're not going to be interested in any of the others. That's just to give you the broader picture of where they fit into the family of phospholipase C enzymes. Now, although all of these catalyze the same thing, they are all regulated by different things. Okay, and the ones which are going to be activated by alpha Q/11 subunits in the on state are the phospholipase C beta family. Okay, so these are the most famous family. Okay, when people just say phospholipase C and they don't put a clarifying Greek letter on the end, generally they will mean a phospholipase C beta family enzyme rather than any of the others. Okay, so let's just draw this out. So, let's say that this here is our alpha Q/11 subunit here, and it's in the on state, so it's got guanosine triphosphate, GTP, bound here. Okay, so let's colour it in vivid purple here. So here's the alpha subunit in vivid purple here. Okay, and it is now going to activate a phospholipase C beta family enzyme. Now these are attached to the underside of the phospholipid bilayer. Well, actually it's slightly more complicated than that. They move between being attached to the underside of the phospholipid bilayer and being in the cytoplasm. Okay, so phospholipase C beta enzymes, they continuously move from being in the cytoplasm to being attached to the underside of the phospholipid bilayer. So there are things within the membrane which they can bind to, basically. So, alpha Q/11 subunits in the on state with guanosine triphosphate bound to them can bind to the phospholipase C beta enzymes that are attached to the underside of the phospholipid bilayer and activate them. Okay, so it's important to understand that the phospholipase C beta family enzyme is continuously moving between being in the cytoplasm and being attached to the underside of the phospholipid bilayer. Now, even when it's attached to the underside of the phospholipid bilayer, it's still inactive. Okay, so just because it's moving around, that doesn't change whether it's active or not. So even if it's attached to the underside of the phospholipid bilayer, bound to components of the phospholipid bilayer, it doesn't mean it's activated, and instead it needs the alpha Q/11 subunit, which is in the on state, to bind to it to activate it. So now this has come along and bound to this phospholipase C beta enzyme. Okay, so it's a protein coded for by one of these 4 genes here, although there are multiple splice variants for some of these genes. Okay, what is this enzyme now going to do? Well, it's going to do what all phospholipase C enzymes do. It's going to break down phosphatidylinositol 4,5-bisphosphate. Okay, so I want to talk about this molecule, phosphatidylinositol 4,5-bisphosphate. So what does this mean? Okay, well, let's take this name bit by bit. So we will start off with the phosphatidyl. Okay, now the first thing actually to say about phosphatidylinositol 4,5-bisphosphate is that it's usually abbreviated to PIP2. Okay, so P for phosphatidyl. Okay, I'll highlight this up. So P here is for phosphatidyl. Okay, the I is for inositol. Okay, and then the P2 is for bisphosphate. Now, if people are being very correct, what they will instead abbreviate this to is PI, and then they'll put brackets 4,5P2, so that they show you which carbons of the inositol ring, and if you don't know what that means, I'll tell you in a moment, which carbons the 2 phosphates actually come off. Because there are other molecules of where you have phosphatidylinositol with 2 phosphate groups coming off, okay, which don't have them coming off the 4th and the 5th carbons, okay. So there are other examples of phosphatidylinositol bisphosphate. However, if you see PIP2 written like this and you don't have numbers in the middle here telling you exactly which carbons these phosphate groups come off, then you can assume that they come off the 4th and the 5th, okay? So only this one here can be abbreviated down to PIP2 like that, or PIP2 as it's often called. Right, so let me show you the structure of this molecule. It is a phospholipid that is within the phospholipid bilayer, okay? And I'm going to go over the page to show you the structure of it. So, we will begin with, what does phosphatidyl mean? Okay, well, phosphatidyl means that within the structure of this larger molecule, you are going to have a molecule that is known as phosphatidic acid. Okay, so I'm going to show you the structure of phosphatidic acid now. Okay, so phosphatidic acid then consists of a glycerol molecule. Glycerol is the fundamental backbone for phosphatidic acid. So I'll colour this vertical line here in green, and this is going to represent our glycerol molecule. Okay, right. So glycerol is the old name for a molecule that would now more correctly be called propane 1,2,3-triol. And again, although propane 1,2,3-triol is much more of a mouthful than glycerol, it's a more useful name because it tells us exactly what this molecule actually is. Propane tells us that we're dealing with a 3-carbon molecule, okay? And the 1,2,3-triol means that we've got alcohol groups coming off the 1st, the 2nd, and the 3rd carbons of that molecule. Okay, so we've got a 3-carbon molecule with alcohol groups coming off the 1st, the 2nd, and the 3rd carbons. Now, to create phosphatidic acid, what you do is you attach onto the alcohol group that comes off the 1st carbon a long-chain carboxylic acid. And you do the same thing for the alcohol group that comes off the 2nd carbon. So these 2 horizontal lines here that I am now colouring in orange here, these are going to represent long-chain carboxylic acids, okay? So these are just carboxylic acid molecules with extremely long tails. Okay, so we've actually seen two examples of long-chain carboxylic acids. We have seen myristic acid and palmitic acid. Those are both examples of long-chain carboxylic acids. Now, the more unofficial name for long-chain carboxylic acids is to call them fatty acids. Okay, so you take these extremely long hydrophobic fatty acids, as they're called, and you attach them to the alcohol groups coming off the first and the second carbons of the glycerol molecule. Okay, and this attachment is done via an ester link. Okay, then to the alcohol group that comes off the third carbon of the glycerol molecule, you are going to attach on a phosphate group. Okay, so this ball here, which I'm colouring in purple, this is a phosphate group. This molecule that I have now shown you, this is the structure of phosphatidic acid. Okay, right. Now, the first thing to say is that there is not just one phosphatidic acid molecule, because I did not specify what the long-chain carboxylic acids in this position and this position should be. These could have been any long-chain carboxylic acids you like, and they don't have to be the same. This one could be different from this one. This could be palmitic acid, this could be myristic acid, okay? But there are loads of other examples of long-chain carboxylic acids present within cells as well. So that means that there are many different phosphatidic acid molecules, okay? Because effectively you have 2 points of variation here. Right, so that's the structure of phosphatidic acid molecule. Phosphatidyl means that we are going to contain phosphatidic acid within our structure. Okay, so we're going to have this structure within phosphatidyl inositol 4,5-bisphosphate. So, the next portion of the name then, phosphatidyl inositol. So, inositol then. We are now going to attach on to the phosphatidic acid molecule an inositol ring. Okay, so what is inositol? Well, once again, inositol is an old name for a molecule that would now more correctly be called cyclohexane. Okay, and you will see very clearly why the old name is preferred to the new name in this case. Cyclohexane 1, 2, 3, 4, 5, 6 hexol. Okay, and again, that's a huge mouthful there, but it's a useful name because it tells us exactly what inositol now is. Cyclohexane tells us that we are dealing with a 6-carbon molecule where you have joined the 6 carbons to form a hexameric ring. Okay, so I'll draw the skeletal structure here. So basically, remember in skeletal structures you don't show carbon atoms, they're implicitly shown by corners. So if I wanted to draw cyclohexane, all I draw is a hexagon like this. Here are my carbons, here, in fact I might colour this in. Here are my carbons, here, 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 and here. Okay, now cyclohexane, that will be all I drew because those are the carbon atoms shown, and then the other bonds that come off these carbon atoms, because each of these carbon atoms still has 2 bonds left, okay, those are both to hydrogen atoms, and in skeletal formulae you don't show hydrogen atoms coming off carbon atoms, so this would be all I showed for cyclohexane. However, we don't want cyclohexane, we want cyclohexane 1, 2, 3, 4, 5, 6 hexol. So we need 6 alcohol groups coming off this molecule, and they need to come off the first, the second, the third, the fourth, and the fifth, and the 6th carbons. So in fact, we're going to have an alcohol group coming off every single one of these carbons, which means every single one of these carbons is going to end up with an alcohol group coming off it, and also a hydrogen coming off it. Okay, now there is a slight subtlety here. You might think, oh wow, how simple, but there is a subtlety here, and the subtlety is optical isomerism. Okay, because if you imagine having this 6-membered ring here in the plane, you can't quite do that, but you can get pretty close. Imagine having that in the same plane, and imagine it's in the plane of the piece of paper. Okay, then the 2 other bonds that come off each of these carbon atoms One will come out of the page towards you, and the other will go into the page away from you. So for each of these bonds, for each of these carbons, you have to decide, do you want the alcohol group coming out of the page towards you and the hydrogen going into the page away from you, or do you want it the other way around? Do you want the hydrogen coming out of the page towards you and the alcohol group going away into the page away from you? Excuse me. Okay, so that is a subtlety with inositol, and overall there are 9 different optical isomers of inositol. Now, we're not going to go through all of the 9 different optical isomers of inositol, and the reason is we don't have to, because only one of them is actually used in cells. The optical isomer of inositol used within biological systems is myo-inositol. Okay, so if you ever hear people referring to myo-inositol and think, what on earth are they talking about? Muscle-inositol? No, all it means is the optical isomer of inositol that's actually used in cells. Generally, people will not use this word, okay? They will just call it inositol, and they will assume that because we are all biologists that we realise that the only inositol we care about is myo-inositol, okay? If you're talking to a chemist, they'll specify it, because they might well work with other optical isomers of inositol, but biologists, we're only ever interested in myo-inositol. Okay, so let me show you then the structure of myo-inositol. So, we'll start with this carbon here. The alcohol group here is going to come out of the page towards us. Now, the way that I show bonds coming out of the page towards us is by having the bond getting thicker towards the end. Okay, so this demonstrates that the alcohol group here is coming out of the page towards us. Now, I'm not going to show the hydrogen going into the page away from us, because remember, we are drawing a skeletal formula here. So we do not show hydrogen atoms coming off carbon atoms, but the hydrogen would be going into the page away from us. Okay, right. Off this next carbon, it's the same story. The alcohol group is going to come out of the page towards us, and the hydrogen is going to go into the page away from us. This next one, again, the same story. So all of them are going in the same direction at the moment as each other. Okay, so this one is also coming out of the page towards us. Then it suddenly breaks. Okay, now one is going to go into the page away from us, and the way that I show bonds going away from us into the page is by these dashed lines. Okay, so this alcohol group is going away from us into the page, and therefore the hydrogen would be coming out of the page towards us, but I'm not going to show that. Okay, on this next one we go back to coming out of the page towards us, okay, like so, and then finally this one here, the alcohol group goes into the page away from us. Now, there is a set way to name up the carbons of a myo-inositol ring. You name this one number 1, this one number 2, this one number 3, this one number 4, this one number 5, and this one number 6. Okay, right. Now that's important it's important for you to know, so that you understand in a moment when I show you the structure of phosphatidylinositol 4,5-bisphosphate, why it is not called phosphatidylinositol 3,4-bisphosphate. Okay, so what we are going to do is we're going to attach this alcohol group that comes off the first carbon to the phosphate group of a phosphatidic acid molecule. Okay, so the first carbon here is going to get attached to the phosphate group via what's known as a phosphoester link formed between this alcohol group here and the phosphate group here. Okay, right, so I'm just going to show this hexagon here, and this is our inositol ring, but remember this is the first carbon here. Okay, right, so there in blue, that's our inositol ring. So that's phosphatidylinositol, and now we want phosphatidylinositol 4,5-bisphosphate. Okay, so what we're now going to do is attach on 2 phosphate groups onto the alcohol groups that come off the 4th and the 5th carbons of the inositol ring. Well, here's the 4th carbon, here's the 5th carbon. Okay, so let's put one phosphate group here, just shown as a blob, and another phosphate group here. Okay, so that is phosphatidylinositol 4,5-bisphosphate, or PI45P2. Okay, Now, this is the reason I've stressed so much about the specific optical isomer of inositol that's been used here. Okay, if I had just drawn that picture, you would now be asking, why is that not called phosphatidyl inositol 3,4 bisphosphate? But understand that that carbon off which this phosphate group comes here is different from this carbon here. Okay, this carbon, this fifth carbon, its alcohol group comes out of the page towards us, okay, and it is positioned in between two carbons which have their alcohol groups going into the page away from us, i.e. going in an opposite direction to the direction which the alcohol group of the fifth carbon goes. Whereas the third carbon, its alcohol group comes out of the page towards us, and it is positioned in between two carbons, one of which has its alcohol group coming in the same direction as the alcohol group coming off the third carbon, and then one which has it going in the opposite direction. So it is fundamentally different from the fifth carbon, okay? Subtly, I agree, but it is fundamentally different. So this comes off the fifth carbon, not the third carbon, and it is possible to tell the difference between the third and the fifth carbon. Okay, so it's phosphatidyl inositol 4,5 bisphosphate, and that is a different molecule to phosphatidyl inositol 3,4 bisphosphate because of the specific optical isomer of inositol that is used within biological systems. Okay, right, so that is phosphatidyl inositol 4,5 bisphosphate. Now, back to phospholipase C beta enzymes. So I'll redraw out where we have got to with our pathway. Okay, so here is our alpha subunit of our heterotrimeric G protein that we have agreed is going to be an alpha Q/11 family alpha subunit. Okay, and I'll just colour that in vivid purple, sticking to convention. Okay, right, and this is now bound to one of these phospholipase C beta family enzymes, whilst the phospholipase C beta family enzyme was transiently attached to the underside of the phospholipid bilayer. Okay, so here is our phospholipase C beta family enzyme. Now, binding of the alpha Q/11 subunit, which is in the on state, and I've just made a horrendous mistake here by not having the GTP bound to the alpha Q/11 subunit. Okay, here is the GTP bound there. Okay, the binding of this alpha Q/11 subunit, which is in the on state, to the phospholipase C beta enzyme, activates the phospholipase C beta enzyme, and it's now going to break down phosphatidyl inositol 4,5 bisphosphate molecules, which are present in the phospholipid bilayer. So I'll draw one here. Okay, so they have the two long-chain carboxylic acids facing into the hydrophobic core of the phospholipid bilayer. Here is their glycerol molecule, here is their phosphate group with the inositol ring here and the two phosphate groups attached to the inositol ring here. Okay, so the polar head is all of this portion, and here is the hydrophobic tails over here. Okay, so here are the long-chain carboxylic acids in orange, here in purple are the three phosphate groups, and here in blue is our inositol ring. Okay, so what's now going to happen is the phospholipase C beta enzyme is going to break down the PIP2 molecule here, specifically PI45P2 here. Okay, and it's going to cut this bond here that's between the glycerol molecule, the third carbon of the glycerol molecule, and the phosphate group of the phosphate group that's attached to the first carbon of the inositol ring. Okay, now that produces two products, one which is going to remain in the phospholipid bilayer. Okay, so let me draw this out separately here and I'll show the two products. So one of the products is just a glycerol molecule with two long-chain carboxylic acids attached to it. So here in orange are the two long-chain carboxylic acids, and here in green is the glycerol molecule, and this molecule is what's known as a diacyl glycerol molecule. Okay, because it's a glycerol molecule with two acyl groups attached to it, which remember is just the portion, well, it's just a carboxylic acid molecule with no alcohol group. Okay, so this is often abbreviated to DAG. Okay, and I'll just go over what an acyl group is, just in case anyone doesn't know. So if I draw the carboxylic acid molecule here, okay, and I'll put an R there for an arbitrary long chain here. Okay, when you attach a long chain carboxylic acid here in orange, okay, so I'll colour this in orange, to the alcohol group of a glycerol molecule here, it's done via an ester link. And in the esterification reaction, what happens is you take the alcohol group of the long chain carboxylic acid, okay, you take the hydrogen of the alcohol group of the glycerol molecule, you combine those two things together to make water, okay, and then you bind the carbon of the carboxylic acid group to the oxygen of the alcohol group here, okay, and that link that you form there is what's known as an ester link. Okay, so this is how you attach long-chain carboxylic acids to alcohol groups on the glycerol molecule. Okay, now you will notice that you have not actually attached on the entire long-chain carboxylic acid to the glycerol molecule. Instead, you have attached on this portion that I have circled in green. You've attached everything but the alcohol group. Okay, now this is no longer a molecule because, you know, this carbon still needs another bond. Instead, it's a group, and this is known as the acyl group of the carboxylic acid molecule. Okay, so this is why this molecule is known as diacylglycerol, because you have attached on two acyl groups onto a glycerol molecule, and for short, diacylglycerol is abbreviated to DAG. Okay, the other product that you produce is the inositol molecule, or more strictly speaking, the myo-inositol molecule, with the three phosphate groups coming off it, one off the first carbon here, and then the other two off the fourth and the fifth carbons down here. Okay, so there are the phosphate groups in blue, and here is the inositol ring in blue. Okay, right, so this molecule now is called inositol, and if you want to be very strict, you'd call it myo-inositol, but we'll just call it inositol along with everyone else in the world. Inositol 1,4,5 tris phosphate. Okay, and for short, inositol 1,4,5 trisphosphate molecules are often abbreviated to IP3 molecules. I for inositol and P for free phosphates. Well, P for phosphate, and then the 3 is for the trisphosphate. Okay, right, so this is an extremely important signaling molecule, IP3. Okay, right, so the IP3 doesn't remain attached to the underside of the phospholipid bilayer, it falls into the cytoplasm, and what it's now going to do is it's going to bind to the cytosolic aspects of receptors known as IP3 receptors, and it's going to trigger the release of calcium from the endoplasmic reticulum. Okay, so let's just talk about this briefly. Okay, so the calcium concentration in the cytoplasm of cells is very, very low. Okay, so let me just draw a little picture of a cell here. So this is our cell here, here's our nucleus. Okay, right, so the normal cytosolic calcium concentration is 100 nanomolar. That's small. Okay, compare that to the extracellular calcium concentration, which is 1.5 millimolar. Okay, that means there's a 15,000-fold gradient of calcium across the cell membrane. There is 15,000 times the concentration of calcium extracellularly than there is intracellularly. Okay, however, there is an intracellular store of calcium. Within the endoplasmic reticulum, which is a membrane-bound intracellular organelle that's often just abbreviated to the ER, you have calcium ions stored. So you do have a store of intracellular calcium ions. There are a lot of calcium ions in the ER. Now, it's a very important part of cell signaling that these calcium ions can be released from the ER into the cytoplasm where they can trigger downstream effects. Okay, and how do you open these intracellular stores? Well, there are two major channels which are involved in releasing calcium from the endoplasmic reticulum. One is the ryanodine receptor, which is extremely important in muscle cells, but we're not going to discuss that here. Okay? The other is the IP3 receptor, and that's the one we're going to discuss, considering that we have just generated IP3. Okay, right. So, at the cell membrane, you've had all of this signaling going on, and you've now produced IP3. And the IP3 is falling down from the cell membrane to the membrane of the endoplasmic reticulum. And in the membrane of the endoplasmic reticulum, you have receptors for IP3. So let me draw one of these here. Okay. So it's an ion channel, so I will draw it as a cylinder in the sort of conventional sense. Okay, and we'll have a little tube down the middle, and basically this is known as the IP3 receptor. Okay, and for short, the IP3 receptor is just abbreviated to the IP3R. Okay, now IP3Rs are not constructed out of a single protein. They are constructed out of multiple proteins that are joined together. Okay, specifically they are tetramers. So, you take 4 separate polypeptides, you join them together to make the full IP3 receptor. So it is not just one polypeptide. Instead, it is made up of 4 separate polypeptides joined together. Okay, so I've highlighted one there in orange. Here is another one being highlighted in turquoise here. And there are 2 more at the back there. Okay, so these individual proteins which you use to build the IP3 receptor, these are known as the IP3 receptor subunits. Okay, so this is called an IP3 receptor subunit. And there are actually 3 different genes in humans which can code for IP3 receptor subunits. Okay, so the 3 different genes are known as the IP3 receptor subunit 1 gene, okay, then the IP3 receptor subunit 2 gene, and then the IP3 receptor subunit 3 gene, or IP3R1, IP3R2, IP3R3. Now, all of these genes have multiple splice variants, but the proteins which they produce, they will all have different sequences of amino acids, but they are similar enough that they can all perform a role as a quarter of an IP3 receptor. Okay, so all of these proteins, all 3 of these genes produce proteins which can perform a role as a quarter of an IP3 receptor. Now, the other thing to say is that these IP3 receptor subunits are actually massive proteins. They're around 2,750 amino acids. Now, that's big. That is very, very big for a protein. And just in case you don't know, alpha stands for amino acids. Okay, so they are big, big proteins, and you're joining 4 of these things together to make a complex, which will be over 11,000 amino acids in total. So these are big things. They're not quite as big as ryanodine receptors, which are so big that you can actually see them under electron microscopes, but they are big. Okay, so the final thing also to say is, how do you assemble 4 of these subunits together? Well, you can either join 4 subunits which are all of the same type together to make what's known as a homotetramer. Okay, so homo means the same, tetramer means 4-membered structure. So this means that the 4 members that you're using to make the full IP3 receptor are the same. Okay, now it doesn't mean that they have to be the identical splice variants. It just means that they have to be made from the same gene. So they have to either be all type 1 IP3 receptor subunits, i.e. have been made from this gene, or they have to all be type 2 IP3 receptor subunits, i.e. they've been made from this gene, or they all have to be type 3 IP3 receptor subunits, i.e. they've been made from this gene. Okay, but we also know that you have heterotetramers, okay, which means that the 4 subunits that you use to make the IP3 receptor are not all the same. Hetero means different. Okay, so you have different types of IP3 receptor subunit in your IP3 receptor. Now, Heterotetrameric IP3 receptors are not well understood at all. We do not know which combinations are allowed, what the stoichiometry is, what the arrangement of the IP3 receptor subunits are. I know many people who are working on trying to find that, but it's not known at the moment. Okay, so I can't give you the juicy information of exactly which combinations are allowed or not. We just know that you have some IP3 receptors where all 4 subunits are made from the same gene, and some where they're made from different genes. Okay, right. The function of the IP3 receptor then, what's going to happen is the IP3 that's been made at the cell surface membrane is going to fall down to the IP3 receptor, and each of the 4 IP3 receptor subunits has a binding site for IP3. Now, you do not need all 4 of the IP3 receptor subunits to bind IP3 in order for the channel to begin the process of opening, and opening is a complicated thing for IP3 receptors, which we're not going to go into in full detail. Okay, we're just going to paint a oversimplified, but a good oversimplified picture. Okay, so the oversimplified picture that we are going to use is that IP3 binds to the IP3 binding sites on these 4 subunits, and that causes the channel to open. Now, it's more complicated than that. There are other things that are needed as well. For instance, calcium is actually extremely important for the IP3 receptor to actually open, but we won't go into these details. Overall, the simplification we're going to take is that when IP3 binds to the IP3 receptors, that leads to the opening of the IP3 receptor. Even if it's not a direct IP3 binds that causes it to open, and in reality it probably isn't. It's other things are needed as well to make it open. But we'll take the oversimplification that IP3 indirectly leads to the IP3 receptor opening. Okay, now what I was saying is that you don't actually need all of the IP3 receptor subunits to bind IP3. I think the minimum requirement is that at least 2 of them have to bind IP3 for the thing to open. Okay, right. So what will then happen is the IP3 receptor will open, the pore will open down the middle here, and now calcium ions in the lumen of the endoplasmic reticulum can leave the lumen of the endoplasmic reticulum by going through the IP3 receptor channel pore here. Okay, and that means that the calcium comes out of the endoplasmic reticulum and into the cytoplasm. So the calcium concentration in the cytoplasm is going to go up. Now, when people say intracellular calcium is going up, what they will often write is something along the lines of this. They'll put Ca2 plus for calcium, then they'll put square brackets, which means concentration of calcium, and then they'll put a little suffix here, I, which means intracellular, and we'd say that the intracellular calcium concentration has gone up. Intracellular in this context means really cytoplasmic, okay? Because if you were thinking about this, you'd say, but hang on, you've just released the calcium from the ER into the cytoplasm. You haven't actually increased the number of calcium ions inside the cell. You've just moved them from the ER into the cytoplasm. Okay, so surely intracellular calcium concentration hasn't gone up. But then you have to remember that intracellular really means cytoplasmic, not endoplasmic reticulum. Okay, so the concentration of calcium ions in the cytoplasm has gone up, basically, which is true. But the concentration of calcium ions in the ER has actually gone down, and they've gone, you know, the ER calcium concentration has gone down by the amount that the cytoplasmic calcium concentration has gone up. Okay, so really, if you were taking intracellular as literally meaning inside the cell, you'd say that the calcium concentration intracellularly hasn't actually changed. However, when we say intracellular calcium concentration, we mean cytoplasmic calcium concentration. Okay, right, so that's the signal that we want to see. That's what we want to study. We want to study this rise in cytoplasmic calcium concentration, and we are going to be able to do this using the Fluo-4 dye. We are going to be able to measure how much the calcium concentration in the cytoplasm goes up in response to stimulating our cell with agonists for G protein coupled receptors, which are coupled to the GQ/11 heterotrimeric G proteins. Okay, so in the next video, what we will begin is our study of Fluo-4 and how we are going to be able to watch as calcium goes up in the cytoplasm of these cells.